observatory built in a controlled environment to study coupled Earth surface processes. And the Earth surface processes that we envision in the Landscape Evolution Observatory are hydrology, geomorphology, geochemistry, ecology, etc., atmospheric sciences. So it's really the entire range of Earth sciences that are going to be represented in this long-term experiment. This is an artistic rendering of the LEO project. You can see it's built within a large greenhouse. We call it Biosphere 2. Biosphere 2 was built in the early 90s. The main purpose of that day was to investigate whether life can continue to exist if you isolate it from the environment and only provide energy through solar light. So all the resources that were used by the people that were living inside the Biosphere 2 had to be recycled from within. There was no air, water, carbon added to the environment. That was completely sealed off from the external Biosphere 1. And then in 2007, the University of Arizona took over the management, the scientific management of this large scale greenhouse. And we decided to do something spectacular and big, and that is the Landscape Evolution Observatory. As you can see it here, it consists of 3 identical landscapes. We call them hill slopes. So this is one hill slope. It has a dimension of 30 meters long and it's 11 meters wide. And the steel container that you see here with a slope of 10 degrees is filled with a 1 meter deep soil that is derived from basalt rock, the type of rock that you can find here around Unam. Basically, it was mined from a tephra mine near Flagstaff, which is in the north of Arizona. And that tephra was ground to fine soil size distribution that is comparable to a loamy sand. So the soil of one of these hills, of all of these hill slopes, is a loamy sand, but it's not a soil. If you would tell this to a soil scientist, he would laugh with you because it doesn't have all the characteristics of a soil. It doesn't have organic matter. It doesn't have clay minerals. It's a very simple primary mineral soil without any structure. So it's basically a homogeneous mixture of particles with a certain distribution, and they're all basaltic glass, basically. I'll explain a little bit later why we chose this approach. As you can see, we've got 3 identical landscapes, so we can do experimentation that is replicable. So if we treat them in the same way, we basically have an N of 3. But we observe, we can statistically analyze, at least prove that what we observe in one, we can also observe in the other 2 hill slopes. So because it's built in a closed environment, we can control temperature, we can control relative humidity, wind speed, we even can control the carbon dioxide concentration in the atmosphere. And therefore, with these large landscapes, having this control over the forcing and the environment, it makes it a unique facility for study. This is a picture taken from the downstream part looking upstream. As you can see, here at the down end of the hill slope, we have a gravel layer. And that gravel layer allows water that has vertically infiltrated into the soil to move laterally out of the soil. We call that seepage flow. And this is a picture of the greenhouse that contains those 3 landscapes. So why did we build this facility? What's the driving research question behind it? Basically, we conceived this Landscape Evolution Observatory to study the coevolution of soils, biota, and resource cycles. And I'll explain this with stepping through this evolution that we anticipate to be able to observe over time. So right now we have a very simple hill slope. This picture simulates that there is vegetation growing, but for the time being there is no vegetation growing. It's just bare soil. and it has a certain slope, certain soil depth, it has a certain topographic convergence, and we know the hydraulic properties of the soil. So with that information, we have very good hydrologic models that can predict how these hill slopes will respond when we rain on it. And the predictions will be of good quality. But as soon as we add water to these hill slopes, we allow microbes to colonize these slopes, because microbes are ubiquitous in the environment, and as soon as there is an environment where they can colonize, they will. But of course, since there is no carbon or nitrogen inside the soil, we expect the microbes that will colonize the soil be autotrophs, so basically microbes that are able to get their carbon and nitrogen from the atmosphere, like plants do. But these microbes will then start adding carbon and nitrogen into the subsurface, and if we allow that to continue for a while, there will be enough carbon and nitrogen in the subsurface that vascular plants will also be able to colonize these initial slopes. So what started off as an abiotic system will, just by adding water and life in the environment, microbes and seeds can be transported in many ways. Visitors can bring them. We can distribute them to simulate seed dispersal in the landscape. We turn this abiotic landscape into a very simple ecosystem. So for the very first time, we have an instrument that allows us to observe how life starts on Earth. We have a fresh lava flow in Hawaii. You come back 100 years later and you see lush vegetation if it is in a humid climate. Okay, the wet side of the big island of Hawaii is a tropical rainforest, and that develops so quickly on new lava flows. What are the processes that allow that life to be so lush on these very simple mineral soils? That is a question that we will be able to address with this equipment. Now, as this continues further, we will see the development of more complex ecosystems. We can have vascular plants competing for water and carbon or nitrogen together with more deeply rooted plants. So there will be interesting dynamics observable at the ecosystem scale. And of course, that will enhance the biogeochemical weathering of the slopes even further. Having those different systems of plants in the slope will also change the subsurface structure and connectivity. By basically root decay will leave macropores in the subsurface. And what started as a very simple matrix flow driven system now becomes a complex system where the water not only moves through the matrix, but probably also through preferential flow paths like macropores. And macropores can be in the vertical as well as in the lateral direction, as suggested by this cartoon here. So when this cycle is completed, what started as a very simple system that hydrologists understand and the models are compatible with no longer meets the assumptions underlying our hydrological models. And we are faced with a system that is too complex to make predictions about current response and is way too complex to make predictions about how this will change over time when we change one of the drivers like climate. So that's the motivation behind the construction of this Landscape Evolution Observatory. And it's built in a way that it basically can address any question that one can conceive in Earth system sciences. And any one of you who is working on a specific topic can pose a question that potentially can be studied in this system. Sample research questions is, can we close the water, carbon, and energy budget? We've never been able to do that with real systems. Here we have the opportunity to prove that we can. What's the role of plants and microbes in controlling hill slope scale weathering pathways and fluxes? That's basically an ecological geochemical question. Can we predict hill slope evolution in a changing climate? That's a climate change question. How does water balance variability influence ecological community assembly? That's an ecological question, etc., etc. So everybody can form his own question, and we can provide the facility to study them. So what do we observe with these hill slopes? This is a map of the subsurface instrumentation that we have available in the slopes. In total, we have about 1,800 sensors and samplers embedded in the soil. Okay, we have, and I'm not sure if you will be able to read it, but we have probes that measure soil moisture and soil temperature. We have lysimeters that allow us to extract soil solution for chemical analysis of soil water. We have soil heat flux plates to measure the soil heat flux across the land surface. We have CO2 sensors that measure the CO2 concentration in the soil air. We have gas samplers that allow us to extract soil air samples from the subsurface. We can send that to an instrument that measures the carbon dioxide concentration or the water vapor concentration, or even isotopes. We have pressure transducers to measure the water table dynamics, the saturated zone. And we have electrode stacks that allow us to scan the subsurface using electrical resistivity tomography. And that technique, it's a geophysical technique, allows us to see changes in the electrical resistivity that are due to either water presence or the mineral changes that are due to chemical weathering, because clay minerals have a higher electrical conductivity than primary minerals. So we will be able to also observe structural changes in the subsurface. Above ground, we have 5 masts hanging from the space frame, very similar to what you would do in micrometeorological studies. They're about 10 meters tall, and then we have at 5 different levels. This is 25 centimeters above ground, this is 2 meters, etc., or 1 meter, I forgot. We have sensors that measure wind speed and wind direction, temperature, relative humidity, visible light or photosynthetic active radiation. At the side masts, we have also 4-way radiometers, so we measure incoming short and long wave radiation and outgoing short and long wave radiation, so from which we can derive the net radiation directly. And then central mass has also a 3D anemometer, sonic anemometer, to measure high frequency turbulence wind variations. So that's a typical array that allows us to measure the atmospheric conditions. And of course, combined with the subsurface array, we now are able to look at land surface atmosphere interactions directly. We also have added remote sensing capability. So this is a 35-meter long rail that is currently positioned above each slope in the central line. And we can hook up a temperature-controlled box, not shown on this slide, that can house these cameras. This is an infrared camera. This is a hyperspectral camera, high resolution, both. So that this one, for instance, can give us an information about the bare soil evaporation because of differences in surface temperature. Whereas this one can give us information about photosynthetic activity of plants through fluorescence measurements. So those are very traditional remote sensing methods that are currently used either from airborne or space. And our facility will also allow the development of remote sensing algorithms that turn this information into something that measures the land surface properties, because we know the land surface properties much better than what you can measure from space and any ground truthing that people have attempted. So even people that are interested in just the development of remote sensing equipment for NASA or any other space agency can come here and develop these algorithms. So we add water to the slopes by using a sprinkler system. This is one of the razors that we have. You have 5 lines, and any combination of them gives us the opportunity to rain on the slopes between 4 and 50 millimeters per hour. For each of the combination of sprinklers, we know the spatial distribution. As you can see, it's not homogeneous, but we know the distribution, so we can account for that if we do hydrological modeling or ecosystems modeling. At the outlet of each slope, we can measure the seepage flow that comes out of the slope when they are wet in 6 different sections along the 11-meter wide seepage phase, and we measure the flow through tipping buckets and electromagnetic flow meters that are barely visible on this slide. The reason why we have those in series is that tipping buckets are very good for low flows, whereas electromagnetic flow meters are good for high flows, and a combination of them gives us an accurate measure of the total flow out of each hill slope in per 15 minutes time intervals. One of the unique features of each of the slopes is these load cells. There are 10 of them. The combined signal of the load cells give us information about the change in total mass of each slope. So when we rain on it with a certain intensity, we would expect the mass to increase at a constant rate equal to the rainfall intensity. So the slope of this line, that represents the increase in mass measured by these load cells, that slope is identical to the rainfall intensity with which we add water to the slopes. Of course, that assumes that we don't have any losses, so all the rain that we add stays in the slope, and that is not true anymore when we have very wet conditions where the water is leaving the slopes through the seepage phase, and there is where we start seeing the deviation from the linear relationship in mass increase. So these load cells allow us to basically validate the amount of rainfall that we add per unit time, and we measure that also by measuring the flow rate in the water distribution system. So we have redundancy in our measurements, which allow us to do corrections if one of the instruments fail, or answer questions related to the accuracy of each of the individual sensors. For instance, if we combine the 500 soil moisture probes that we have in each slope, and this is a picture of one of those sensors, it's a 5TM decagon, and we can combine all these 500 measurements and interpolate, we can also reconstruct the increase in mass derived from the volumetric soil moisture. And as you can see, there are some discrepancies with the expected linear increase that are due to potentially errors in the sensors, or issues with the interpolation methods that we've used. So that is an example of how we can combine measurements and look at accuracies and redundancies. As I mentioned, we have 500 soil suction lysimeters embedded in each slope, and they're connected to via tubes. Initially, we connected them to these syringes, and when we pull a vacuum in each of these syringes, then slowly water will seep into the syringe, so that soil water that was in the slope at that location moves into the porous cup of the lysimeter and then is sucked through the vacuum into each of these syringes, which then allows us to collect those samples for chemical analysis. You can imagine 500 for each slope, 1,500 in total. You need an army of undergrad students to get that information. And basically, when you want to do that for one slope and pulling all 500 syringes, it takes a day. It takes a day to get that information. So we have the best resolution we have. I mean, depending on, of course, how many students you can employ, but that is limited. So basically, what the best we could do with this system is that we would have a daily sample across the slope. In order to avoid over-frustrated undergrads, we invested in a vacuum system. So this is an acrylic box. You can see those holes. Those holes will contain cups that allow us to collect samples from a vacuum drone within the chamber of each of these acrylic boxes. And so we can set a specific vacuum for each box, and we've got like 11 for each hill slope, depending on which sector of the hill slope they are sampling. If they're sampling a drier part, we can actually pull a larger vacuum than if we are in a wetter part, which would require a smaller vacuum to get some samples. We can also measure, since we measure the water potential combined with the water soil moisture content, we can also set the vacuum equal to the water potential. So we know exactly how much vacuum we need in order to extract a soil water sample. This allows us to get temporal resolutions of a few times per day. So that's a real advantage. Recently we added laser spectroscopic instrumentations that allow us to measure the isotope concentration of liquid and water vapor. And this is a picture of the off-axis integrated cavity output spectroscopy instrument of Los Gatos or OA ICOS. This instrument allows us to grab a sample automatically from the seepage water and analyze it in real time. So basically how it works is we have a peristaltic pump connected to the seepage flow that pumps water into this autosampler at this device, and then the syringe takes a sample from one of 3 tubes that are here, and then injected into the instrument where it's vaporized, totally vaporized, and then it measures the isotope concentration of water. So that basically is deuterium and O18 by using laser light, which is bounced back and forth like 10 kilometers in total, and then the spectrum is analyzed. So it's a very simple method, but very elegant to look at the isotope composition of water as it seeps out of the hill slope. So now imagine that we have an experiment where we rain on it and we have added deuterium to the rainwater, large amounts of deuterium. Typically, the water source we use, the deuterium concentration is negative 60 per mil. We can spike it up to 1,000 per mil. So there's a huge orders of magnitude difference between what the background is and what we have added. So there's absolutely no doubt when we grab a sample from the seepage and it has a, say, deuterium concentration of 200, that is water that we've added during that event. So because that large contrast that is not achievable in nature, we can do a much better job in tracing how the water moves through the hill slope. And that is important not only for hydrologists, but it's also important for geochemists, because the time it spends in the subsurface is the time it can weather the minerals and reach an equilibrium, maybe over supersaturation, which allows for precipitation of secondary minerals, clay minerals. So they have never been able to do that type of analysis or observation in real systems, and we will be able for the first time doing it here at scales that are representative for real hill slopes. This is a picture of a dual laser quantum cascade spectrometer developed by Aerodyne. This instrument measures directly the water vapor or whatever vacuum system, vacuum pump. We can sample each of those gases analyzer can analyze isotopes of both water vapor and carbon dioxide, C13. So we will have a direct measurement of where the carbon is in the subsurface. If we, for instance, run an experiment where we add carbon-13 to the atmosphere, imagine there are plants growing on the slope. They are photosynthesizing. They take carbon dioxide. Some of it will be labeled with C13. And then when we find it in the subsurface, that means that that carbon has made its way through the plant. It's not stored somehow in tissue, but is extruded by roots into the subsurface. So the tracing carbon as it moves from the atmosphere through plants into the subsurface and therefore provide a source of protons to the chemical weathering reactions of the subsurface is again something we know exists, but nobody's ever been able to observe it. The other cool thing about this is that we measure isotopes of water in the vapor phase. There is, we know from observations and from theory that the water vapor phase, the isotope concentration in the water vapor phase in the soil is in equilibrium with the liquid water that is present in the pores of the soil. So by analyzing the soil air, we actually measure the isotope composition of the liquid water. Now, before this, the only way we could do that was drawing a sample, a liquid water sample from the soil with our lysimeters, which is possible in wet conditions, but in dry conditions it's virtually impossible. So for the first time, we will be able to not only follow the water molecule as it moves through a hill slope in wet conditions, but we will also be able to say where that water molecule is under dry conditions. And basically, then it's where it's stuck. It's not moving, but we know where it is and how it's evolving. So that will add a whole new spectrum to the answers that we can, the questions that we can answer from all the disciplines, from hydrology to geochemistry to ecology. All right, so enough introduction. I hope this will work. So let me show you a video that we've put together based on the soil moisture data from the first experiment that we performed on one of the slopes in 2013. So what we will see is this is a slope, and you can see we've tried to cut it open, so we'll see some vertical slices as well as the surface here. That will allow us to observe how water moves through the slope as we add it through the sprinkler system. So we rained on this slope at an intensity of 12 millimeters per hour at a constant rate. Our models predicted that after 24 hours, the slope would become in steady state. That means the amount of water that entered through rainfall is equal to the amount of water that leaves through seepage. And so there is no internal storage change after steady state. That's an experiment that every hydrologist just dreams of, because that basically is a step response to steady state. And if you have some system engineering background, you know that the step response to steady state tells you everything you need to know about the dynamics of that system. And so that's the reason why we executed it. We also wanted to do that with isotope labeled water so that we can also look at the breakthrough of the tracers, which is different from the breakthrough of the water. Because the flux of water is driven by hydraulic gradients, whereas the flux of tracer is driven by the velocity of water molecules. Okay. All right, so this is the rainfall event, and this red line is the seepage hydrograph. So it's after so many hours, it started to seep out. So the first hours we won't see any seepage; it just gets stored in the subsurface under unsaturated conditions. And then what we see here is the increment of storage, and just for the time being, focus on the black line, which is measured by the load cells. So it increases linearly, then it bends a little bit because we are losing water now through seepage, and then it reaches maximum when we turn off the rain, and then it starts losing it mainly because of seepage. Oh yeah, one more thing before we start the video. We have color-coded the response in light blue, medium blue, and dark blue. Light blue means the initial soil moisture, pretty dry, about 10%. Medium blue is unsaturated but wet, okay? And then dark blue is saturated, about 40%. So we start raining on it. You see the wetting front. The whole surface is now wet. You see the wetting front propagating more or less uniformly. Then you see the development of a water table, and you see this water table ridging here in the central part of the hill, so because of the convergence. And what we see at the end of the experiment is that this part is saturated. That means that here and here, the water table has reached the surface. What you think will, well, let's watch it one more time. So here we see the uniform propagation of the wetting front, indicating that we are dealing with homogeneous soil. Now we see the development of a groundwater table. Here we see the ridging causing saturation in the middle of the slope, and then it expands upslope. That's the result. So because of surface saturation, we got gully erosion. It started here in this part where we saw the water table reaching the surface, and the water then started flowing overland and created this beautiful gully with meandering and everything. But we were not expecting this. It was an unexpected result. Remember, our models predicted that we would reach a steady state without getting this saturation. We never reached steady state during this experiment, which lasted 22 hours. Our model said in 24 hours we would reach steady state with no saturation. So there was clearly something wrong with the model. So when you plan something big, expect failure. And the lesson you learn from the failure is that if it wouldn't go wrong, we wouldn't have learned anything. So I was shocked when I saw this, as you can imagine, and didn't sleep for 3 weeks or so. But then I started realizing, yes, this is an opportunity to actually challenge our models. Why were our models not good enough to predict this? Is it because of the internal structure based on the Richard equation, or is it because of the parameterization, the hydraulic properties of the soil? Because we have measured them, but we measured them at core scale. Here we need hydraulic properties of the entire hill slope. And there's a difference, we know that, but here we have the proof that how different they are. All right, there are a couple of papers that came out of this experiment, and if you're interested in the details, I encourage you to look them up. I just want to give you an example of collaboration between hydrology and geochemistry, because that illustrates the importance of this instrument for interdisciplinary research in ways that has never been done before. So this is, I'm going to show you some results from an NSF-funded project, which is a collaborative research between the University of Arizona and Johns Hopkins University. Basically what we were looking at was, when we add water to the soil, can we observe the chemical weathering? And how much chemical weathering will there be? And where will that chemical weathering take place? Because if we have a certain location that weathers faster than another location because there's more water there, then we also expect that place to start forming clay minerals earlier into the experiment. Clay minerals will change hydraulic properties of the soil. It will have a higher water holding capacity. It will have a lower hydraulic conductivity. So this interaction between hydrology, the evolution or co-evolution of the hydrology and the chemical weathering, that is something that we can observe here. And for those of you who were in my talk yesterday, now understand why this is an instrument that can study catchment co-evolution from all different aspects. Anyway, so what we are looking at this graph here is in time. We're looking at the carbon dioxide concentration measured by our subsurface sensors. So these are the embedded sensors that measure the soil, air, carbon dioxide concentration. And the different colors show the different depths. So the blue is 5 centimeter, the red 20, the green 35. Okay? And the reason why we have differences in CO2 concentrations, at 5 centimeter we've got about ambient, a little bit lower than ambient. Ambient would be 400. And then we have lower and yet lower and deeper depths. And that basically is probably a diffusion limitation of carbon dioxide from the atmosphere that is not diffusing into the soil rapidly enough. All right, and then at some point, and then here we look at the, here we look at the soil moisture in volumetric water content. So initially we had about 10%, and then at hour 12 or thereabout, we started raining on the slope. And as you can see, the 5 centimeters CO2 concentration plummeted immediately as we add water. Then followed by the red and followed by the green. And simultaneously, we see an increase in soil moisture that reaches an equilibrium at around 25%, followed by the red, followed by the green. So the changes in carbon dioxide coincide with changes in moisture content. So why is that? When you add water to the subsurface and there is carbon dioxide in the soil air, that carbon dioxide will diffuse into the liquid water. Okay? It will dissolve into the liquid water. It reacts with the H2O, H2 and CO2, to form carbonic acid, which delivers protons to the chemical weathering and basically leaves bicarbonate in solution. So that's what we call inorganic carbon or dissolved inorganic carbon. That's mainly bicarbonate. So that basically is the carbon cycle that is related to chemical weathering of primary minerals. So basically what we see here in action is chemical weathering. And the surprising result or observation from this is that how quickly it starts, how quickly these processes start. Okay, this is an indication but not a proof of chemical weathering. So for a proof of chemical weathering, we need to look at the carbon balance. Okay, we need to see during an entire event, so now we are not looking at 24 hours, but we're looking at 120 or 100 days, we look at the total inorganic carbon that is stored within the slope. The initial carbon that was stored in the slope was based on early measurements, based on the initial conditions. As you can see, we have a slight increase. And then this rain event, we basically the increase here between the initial rain event and this big rain event is the slow recovery of the loss of CO2 in chemical reactions by diffusion of carbon dioxide from the atmosphere into the slope. So we reach a new level, and then after the rain we see again a very sharp decrease in carbon, which is basically the transformation due to chemical weathering and a very slow recovery. And then again, after rain, a drop. And when you look at the total carbon balance, we basically have a perfect fit between what we add, which is basically carbon dioxide dissolved into the rainwater, and what we export, which is the bicarbonate concentration of the seepage flow. And then, of course, the difference is the dynamic storage of carbon in the subsurface. So this also proves that you can derive chemical weathering denudation by looking at bicarbonate concentrations in the stream flow at some spatial scale, which basically people use in order to estimate the rate of chemical weathering of landscapes. So that's just an example of how hydrologists and geochemists can work together and discover new processes that were hypothesized about, but never really shown in the accuracy and the precision that we can reach with this instrument. We also are fortunate enough to work internationally or collaborate internationally with groups that have similar interests and similar equipment. One example is the Ecotron facility at Île-de-France, which is located near Paris. There they have 8 of these mesocosms, so these cells, where you can put a cylindric device that can contain whatever you want to study. And this cell can be environmentally controlled to a precision that is hard to imagine. 0.1 Celsius, 0.1 relative humidity, CO2 concentration, whatever you want. Much better than what we can control in Biosphere 2, because of course it's much easier to control a small volume like this compared to the large volume of Biosphere 2. So basically what we have is a chain of small scale, very highly controlled laboratory experimentation here in Ile-de-France near Paris. And then we've got Biosphere 2, which is intermediate between what you can find in the real world, in the real landscape. So Biosphere 2 will be able to translate what we measure here at very high accuracy to what we measure in the field with very low accuracy and lots of uncertainty. And so basically what we did, we've shipped a container of basalt soil that we used in Leo to Paris. And for some reason it made it there. I thought it was never going to go through customs. Who ships a container of black soil to Paris? But apparently it didn't raise any flags. And so the soil was then stacked or packed into these cylinders that are about 1 meter deep, same as the Leo soil. And then from the side, we could attach all kinds of sensors. Like, for instance, this is an example of a gas probe that we also use in Leo. So this allows you to extract soil air at this depth. This is an example of the soil. And then once we had packed all these cylinders, we could use a control that was basically bare soil, and then different plants. This is, I think this is mesquite. So that's a typical tree from the southwest. It's a nitrogen fixer. This is a herbaceous nitrogen fixer, and then this is a grass, and different stages of development. And the idea is here to see how are plants going to affect the chemical weathering rates that we already observed under bare soil conditions. So any rate that is higher, that we measure higher than the control, is the result of the plants that are present there at that location. So this we can do at very high accuracy, and we can also do this in very high replication. So we have much stronger statistical power to analyze the data. But we can then translate that information back into Leo and make predictions about how the full ecosystem will affect the evolution of the landscape. So that's just an example of how we try to collaborate internationally to cover more ground than what we can do with the Biosphere 2 Leo. Okay, what's in the future for Leo? So right now it's bare soil, and it's mainly hydrology, geochemistry. The questions that we are asking, and atmospheric sciences, the land surface, atmosphere interactions under bare soil conditions. That's also something we can study in great detail. But then ultimately, in a couple of years, we will start growing plants. We probably will provide a seed bank of local annuals. Oops. Sorry. Oh no. Those of you who just wanted to see what I was talking about, can have a quick overview again. All right, so we will provide a seed bank of local annuals, possibly some woody species that will grow on the slopes, and we are currently doing experiments to see which species can grow under what conditions, and then ultimately we will be able to ask questions about maybe if we add invasive species or we add shrubby plants, how is that going to change the ecosystem dynamics of initially something that was more native to the region around Biosphere 2, which is located near Tucson in Arizona. So when you would visit the Leo in, say, 5 years from now, I hope this is what it will look like, a little bit more interesting than a black surface that reminds you of the surface of Mars. By the way, we got lots of attention when the Martian came out, because the soil in Mars is basalt. And what the guy was doing was growing food on abiotic soil by adding carbon and nitrogen. And basically that's what we're doing here. but we're doing it under natural conditions, not with an agriculture background, but more an ecosystems, natural systems background. Maybe we can ask questions about what difference between C3 and C4, routing depth, phenology, etc., things that ecologists are interested in. So just to summarize or to conclude this talk, I hope I could convince you that Landscape Evolution Observatory offers unique collaborative research opportunities for earth scientists. So I don't know your background, whether you're all hydrologists or a variety of backgrounds, I hope, so that you each can think about what options or opportunities this instrument presents. And we really run this facility as an open infrastructure. We consider it like a telescope in planetary sciences. Not everybody has a telescope, only a few on Earth, but every planetary science can borrow time on a telescope, if it fits a certain context for that particular instrument. And that's the same way we want to treat this instrument, as a telescope to observe Earth system processes. And if you propose your own experiment to address your own research question and it fits the overall structure of the instrument or of the experiment, the long-term experiment, then we definitely will be able to implement it. If we have some limited funding, we probably will be able to find some funding for it. Through interactive cycles of large-scale experimentation, numerical modeling, I didn't talk too much about it, but you can imagine that all the data now allows us to develop models and test models and improve models. We basically can use models to make predictions about a certain experimental outcome, like we did with this steady state experiment, then run the experiment and then fail and go back to the model and say, what's wrong? Why didn't it work? So, and we can do that for hydrology, but also for geochemistry and ecology. And believe me, if you think hydrological models are bad, think about the geochemical models and the ecological models. They have much more challenges than we have. So we can improve our understanding of coupled Earth surface processes. And of course, a vast amount of data can be mined for improving parameterizations critical to accurate Earth systems modeling. So basically, all the land surface modeling that is taking place for climate change predictions, they are based on parameterizations that may or may not be representative of what's going on. And here we can actually test those parameterizations in much more detail than has ever been before. There's just a couple of things that are possible with LEO. So with that, thank you for your attention. So questions? Yes.
Are dataset public?
Yes.
We can access and processing the information?
Yes. Yes. So we have an online database that collects the data from the sensors in real time. And so it's every day, it's up to date. And if you want access to some part of the data or all of the data, you just have to contact me and explain a little bit what you want to do with the data, and what's your question, what type of data you need, and then I can give you access to the database. The only thing is you have to sign like a data sharing agreement, which basically mentions that if you are publishing this, you of course have to recognize the source of the data, and possibly collaborate with some of us if you want, but that's not needed. Yes.
Hello. Have you seen any mineralogical changes inside the cracks? I mean, geometrical forms or whatever?
Yeah, very good question. We have seen through soil solution chemical analysis that certain locations show supersaturation in certain elements, which suggests that there may be clay mineral precipitation at those locations. Of course, that's just indirect observation. What we need to do is go to those locations and take a soil sample and then do the mineral analysis to see if indeed those minerals have been formed. We have several other facilities where we can experiment with the same soil and where we can do much more frequent soil sampling, destructive soil sampling, where we can address that question. So far, we haven't done the mineralogical analysis to positively confirm that we have a new mineral formation, but it is very likely. And it's also coming. I mean, in a couple of months, and we will have the results. Thank you. Adrian.
Thank you very much. Job you're doing there. I was just wondering, as I saw the introduction of the slopes. First of all, what do you think? How representative are these slopes from what you get in the real world? Because obviously you have the bare soil with a thickness of 1 meter.
Yeah.
So I imagine that this deepness or this depth at some point interacts with the flow that you are putting on top of it, no, with the rain. So does this interaction of the depth deviates your results, or how did you define that depth?
Okay. I think there are two questions. So one, how representative are these landscapes? And then second, why did you choose 1 meter? So to answer the first question, we should stress that these landscapes are artificial landscapes. They are not natural landscapes. And we deliberately try to make them as artificial as possible. But they contain all the processes that are present that shape the Earth's surface. Atmospheric interaction, hydrology, geochemistry, ecology, all the things that shape our landscapes. The processes interact in this artificial landscape in ways that are similar to real landscapes, but we can never claim that what we are observing is a true, real landscape evolution, because we've basically created initial conditions that are not realistic. Lava flows don't deposit a loamy sand soil on a 30 meter long, 11 meter wide, 1 meter deep platform. It doesn't, right? But I'm still convinced that we can learn by observing those couple processes, that we can still learn about nature, or at least what we've learned here, confront it with what we've observed in nature, and then see whether it confirms our observations or it contradicts them. And then that's also interesting. Why is it different? And we already saw differences in the chemical weathering that it would have been too much detail to talk about, but I'm happy to continue this conversation offline. Why 1 meter deep? Okay, imagine it's 2007. You bring together a group of 30 people, 5 hydrologists, 5 ecologists, 5 geomorphologists, 5 geochemists, 5 atmospheric scientists. And you ask them, what's your ideal design of whatever initial ideas we had about what LEO would be? We knew it was we didn't even know it was going to be replicated. Imagine what every group would come up with, their own research question, right? I, as a hydrologist, I wanted 3 different slopes, convergent, divergence, parallel. I wanted to change the slope angle with hydraulic lifts. Ecologists, they wanted identical slopes. They didn't want convergence. They wanted it as simple as possible, even preferably flat. They didn't care about lateral redistribution. Geomorphologists wanted erosion. This was going to be an erosion experiment, and so on, and so on. And then we said, okay, we cannot come to a compromise. If everybody sticks to his grounds, we cannot come to a compromise. So we had to start building consensus. And the way we built consensus was through modeling. So everybody brought his own model. And then we asked questions of how deep the soil would be. Well, what is important about the depth of the soil? If it's too deep, it will take too long for it to saturate. If it's too shallow, it will all saturate once in a while, immediately, and flush away. So there is this optimal depth that, from a water perspective, a water holding capacity perspective, must exist. And when we ran our models, we experimented between some possible limitations, and we came up with 1 meter. So that was how that 1 meter was chosen. And we did that with many other things. The basalt soil was chosen because it weathers the quickest on Earth. Even though it is not the most ubiquitous bedrock on Earth, it accounts for 30% of the geological carbon cycle. As you know, the geological carbon cycle is extremely important for the Earth's climate. So understanding that is very important. So geochemists wanted to work with basalt. And they didn't want a real soil. They wanted a simple soil so that they could look at the evolution. That's something that I never thought of as a hydrologist. I thought, okay, where are we going to get soil from? These guys said, no, basalt, primary minerals, nothing else. Then we can look at how that develops. So that's how it came about. And as we developed the design of LEO, we had to develop also a common language and understanding of each individual's driving questions. And what came out were much more rich questions, for me, more rich questions that I never had asked before. Because now I'm interested, the geochemical weathering affects the hydrologic properties of the soil. I want to know how that works. Because now I may be able to say at that location, 1 million years of evolution, maybe the soils are such and such. And that's important for our hydrologic models. So that's the philosophy behind the design. Thank you. Yes.
Hi, Doctor. Thank you for being here. Bienvenido.
I'm very happy to be here.
You replicate artificial landscapes, which means a lot of resources. And that's why, going back to the concept of Pidon that you mentioned yesterday, do you think that it's, or there are any differences of studying the Pidon and modeling the Pidon with hydrological behaviors and do it several times or many times and considering the geomorphological changes and then try to construct a landscape and so make a general hydrological behavior? And what would be the difference between doing this and modeling the whole landscape?
So you're saying what's the difference between trying to understand at the Pidon scale, which is basically vertical processes?
Yeah.
Compared to and different?
Yes, and do it many times and just try to construct a landscape.
Yeah, okay.
And in terms of reliable data, pairing with the modeling of a whole landscape.
Yeah. Well, I think we can do that at LEO. You can consider, if you take a 1 by 1 meter surface and you consider the vertical, so it's a 1 meter deep Pidon, it's a cubic meter. That will contain enough sensors and samplers to study that Pidon. Now, if it's at the top of the slope, that Pidon will only be affected by vertical fluxes, very limited lateral fluxes, because that part is probably never going to saturate. If we saturate that part, we are going to flood the slope again. So that's 1 Pidon, only vertical flux. Now I can take another Pidon somewhere in the convergence zone where we saw the Gully formation. That Pidon is not only affected by vertical fluxes, but also by lateral fluxes. And I would say even more by lateral than by vertical fluxes. So we expect that Pidon to have a very different evolutionary trajectory than the initial Pidon. So we can actually break it up in, 300 Pidons, so to speak, 330 Pidons, and look at how they evolve differently. That's basically what we're doing. Now, there is also a need to do that at the whole landscape scale, because imagine we have an ecosystem growing there. It's 330 square meter ecosystem, a variety of species. With our load cells, we will be able to measure the whole ecosystem evaporation rate or evapotranspiration rate. Normal lysimeters, they do it at a Pidon scale, which is nice, but we will be able to do it at the whole ecosystem scale and how these different species interact in order to sustain a certain transpiration rate. So I think there is a need for both scales. That's also why we do the ecotron experiments. I hope I answered your question. Okay, thanks.
I don't know if my question is like out of topic, but I want to know about a urban catchment. I don't know. Is it possible that this model, can you use it in a urban catchment, or maybe the knowledge that you are getting here? And because I understand that this model is only for like a natural environment, but what about if you have it for I mean, like the interaction between the natural environment and with the urban environment?
Yes, excellent question. And certain elements of urban hydrology or urban catchments will definitely have some relation to what we do. Not all urban catchments, not every square meter of an urban catchment is paved. There are natural soils, there are ecosystems. So those parts of the urban catchments that will definitely have relationships to what we do here. You're absolutely right. We are trying to mimic a natural system. No human involvement other than we turning on the rain. But if you're interested in, say, urban hydrology and the effect of paved area on the flashiness of urban runoff, obviously this is not the type of model that you will use to investigate that. Then you would probably need a different experimental facility, which is probably easier to build than this one. Okay, thank you.
Other questions? Okay, I have to.
Okay.
So have you solved the issue or the problem you found in your model for not predicting overland flow?
I guess somebody's trying to tell us, get out of here.
Okay.
Turning on the light. Yes, it's a good question. And we actually have written a couple of papers on it. So we used that hydrologic model to make the predictions. We were wrong. And of course, we wanted to find out why we were wrong. It turns out that the most likely explanation why we failed to predict this is that this huge experiment that we implemented, 22 hours of 12 millimeters per hour, that's a lot of water, right? That caused massive movement of fine particles in the subsurface. Imagine we just had put the soil on top. We had this fine mixture of particles. Some of them ended up at the downslope end of the slope, clogging basically the pore space. So the reason why we think that was what's happening is that the only way we could reconstruct the observations was by having a hydraulic conductivity at the bottom slope, at the seepage phase, which was lower than the rest of the slope. And why would it be lower other than compaction and movement of particles, fine particles clogging up the pore spaces? Now, when will we know? When we dig out the soil. So we have to wait 10 years. Or we run other experiments that would be able to reproduce that as well. And we have actually done that with the mini Leo.
So in natural landscapes, this clogging effect is likely not to happen.
Exactly. This is an example of why it's not a natural system. The clogging would never occur, right? Or has occurred so many years ago that it doesn't really matter anymore. Yes, we have to separate the things that can be translated to nature things that actually are very specific to the instrument itself. Yeah, absolutely right.
Okay. And the second question is, what so far, what has been the main discovery found out, like just in hydrology so far at LEO?
The main discovery, well, I don't think we have discovered anything yet in hydrology, and actually I'm not expecting that. What I'm expecting more is that we will discover in the interface between disciplines. So to give you an example, in the interface between hydrology and geochemistry, we measured the weathering rate at the hill slope scale, and what we were expecting was, given the conditions, that the weathering rates would be comparable to weathering rates measured in laboratory settings. And there's a lot of literature on weathering of basalt in laboratory, columns where they have flow through it, things like that. What we found was that the weathering rates in the LEO were actually similar to the ones you measure in the field, which are derived from river bicarbonate exports and the chemical denudation of the landscape. So why is that? Why does this very simple basalt soil, initially pure primary minerals, why doesn't it weather as fast as laboratory soil? And what we discovered was that as you add water to the soil, which is needed to start the chemical weathering reactions, what you also do is you basically block the pore space by adding water to the pore space, and you limit the diffusion of carbon dioxide into the slope. Now you need the carbon dioxide to sustain the weathering rates. As you could see when you add water to the slope, we basically deplete the carbon dioxide very rapidly. It all diffuses in the soil, forms carbonic acid, bicarbonate, etc. That doesn't happen in reality, but it's a limitation to the chemical weathering rate. So the reason why we don't measure lab weathering rates is because of a diffusion limitation of carbon dioxide from the atmosphere into the subsurface. Now, what happens in real world is that microbes are present in the subsurface, and microbes, they turn on when it's wet, and then they start respiring, and they produce CO2. So they basically are a source of CO2 in the subsurface, and they keep the weathering going. But in real landscape, you still have low weathering rates. So why are they low? Well, that's a question that we probably will be able to answer in a couple of years from now, when we have more microbes in the subsurface. So is that a discovery of importance? No, because diffusion limitation doesn't happen in natural soils, because you always have microbes in the soil. Only mine tailings maybe. And so for artificial systems, we can actually learn a lot. Or geoengineering solutions to carbon dioxide in the atmosphere. Spread basalt all over the Earth's surface, and you sequester carbon dioxide by weathering. People have actually proposed that. So we can help them in designing those systems. But once we have more microbes and it becomes a more natural system, we actually will be able to address the question of why are the weathering rates so low at landscape scales. Right now, there are several hypotheses, but nobody has ever been able to prove those hypotheses. So I expect those to become more and more frequent.
At the beginning, you had 3 identical big size experiment facility. I don't know if you mentioned it, but what will be the difference between the 3 greenhouse? You have started your experiment, so is there any difference in? I suppose you will compare something between the 3. Did you mention it, or is this a top secret, or where it is? What is the difference between the 3?
Can I refuse to answer? No. Yeah, that's an excellent question. So basically, we build those 3 slopes identically. Everything's the same. Now, everybody who has ever packed soil in a container knows how difficult it is to do that 3 times. Now imagine 330 cubic meters of soil, packing it in an identical way. So we are hoping that the packing was done uniformly across the 3 slopes, but we don't know. So we haven't really done an experiment where we force each of the 3 slopes at the same time, the same amount of rainfall. We haven't done that yet. Every time we rain on it was like individual hill slopes. But we have done experiments on all 3 hill slopes, and we've compared the results. And as far as we could compare them, but because always slightly different conditions. And yes, one of the slopes behaves differently hydrologically than the 2 others. And it's a dramatic difference. The water balance is the same. The runoff coefficient at the end of the experiment is the same as the other slopes. But the way it contains water, retains water, is very different. 2 slopes, they release the water very rapidly. You have a flashy hydrograph rise and a quick recession, whereas the 1 slope has a very slow rise and a very slow recession. Why? We don't know. I have a student working on it right now. The only reason it can be, I mean, there's no other reason I can think of, is changes in the hydraulic properties due to changes in the way that slope was packed. And so can we now, by inverse modeling, can we correct the hydraulic properties for that slope so that we know at least how different they are? That's basically what we are currently doing. Yeah. But there you go. It's the first surprise already. Yeah. Now, when we look at the geochemical response, the way the soil weathers, we don't see any difference. So from a geochemical perspective, they're still identical. The question is, how is that difference in hydrology now going to affect all the other evolution? So it may be that before we actually started the experiment, we already have lost n of 3, and we are left over with n of 2. That's pretty bad news.
Okay, no more questions? Well, thank you very much again, Peter, for this wonderful talk.
Yeah.
So we'll give you again a second acknowledgement, and also we do have some books for you. Yes. Coffee mug.
Oh, great. I love coffee.
Yes, 3 books were edited, well, actually done by this.
Oh, okay.
One about the cathedral,
Okay.
rescue, and then
nice.
about the Chilean earthquake, some
Una.
researchers, also history of Mexican engineering. Hope you'll.
Thank you so much. I will. And thank you.